Good morning, good afternoon, good evening, good day, hello, and welcome. 2700, where today we are doing another exciting ride through a phylum that perhaps you haven't heard of before, so this is my opportunity to proselytize to you, to evangelicalize, and to talk about the Xenacoelomorpha, which is another good swear word I offer up to you, if stubbing your toe. And so we're going to be working in here, and before we get to that, we're talking about things that are in Brusca, the 3rd edition, chapter 9. Where have we been? Well, we were on another exciting ride with Conan O'Brien in the Xenaphores. We talked about characteristics that, the odd characteristics, the 8 teen rows, the apical organ, the statocyst, all of the coloblast, all these things that they can live and be pelagic predators or benthic filter feeders, that stress makes them do crazy things. They can fuse together and act as one organism. They can go backwards through development if they're stressed. And then our understanding of how they sit in the animal phylogeny, how that has blown up since I took this course a million years ago in the last century, and has reframed how we understand the nervous system, the evolution of the nervous system, and the through gut. That in each case, each of those really fundamental traits may have evolved independently more than one time. So today we're talking about, we're going to begin talking about the bilaterian. We're going to do it, which is the taxon, bilaterally symmetrical, the taxon that we are part of. And this group, the Xenacoelomorpha, exemplified with the pictures of different species of Xenoturbella, which is a lot of the narrative, kind of the story that we're going to weave about. We're going to talk about characters, key characters, how this taxon presents some very key questions about animal phylogenetics and our understanding of evolution within our own taxon. And we'll talk about the particularly convoluted taxonomic history of the Xenoturbella, about where we think its final taxonomic position may be, the consequences to that, and different discoveries. And we're going to take you literally out in the ship and share with you some discoveries of novel Xenoturbella. And so, shall we begin? So let's begin. The learning objectives I have today are to think about and understand, to appraise, synthesize, and interpret examples of different hypotheses regarding bilateral, the evolution of bilaterians, to do the same kind of key fundamental skills inside or outside of invertebrate morphology and evolution with Xenoturbella, and understand and interpret the positions and the consequences of those positions within the animal phylogeny. So to start out, if we're talking about bilaterians, remember we've talked about a couple of phyla, the sponges and the placozoans, that have no symmetry at all, the cnidarians that have radial symmetry, and the ctenophores, which have this intermediate kind of biradial symmetry. So a bilaterally symmetrical organism, go and look in the mirror, it's you. This is this thing that where there is a left-right mirroring over that sagittal plane, okay? That within the bilateria, we see an increase in these kinds of traits. So synapomorphies, that we see an increase in terms of cephalization. Zombies have no interest in cnidarians, ctenophores, placozoans, or sponges. There's no brain to eat. So an increase in cephalization, the frequency and the utility of cephalization. We are triploblasts. So we have an endoderm, an ectoderm, and a mesoderm. We're characterized by a through gut, and we have excretory organs, because as we get bigger and bigger and we're producing these toxic byproducts of metabolism like ammonia, we need different ways to get those out of us and vent them into the outside world. So we'll be talking about Brusca chapter 9 from the 3rd edition. That's the Xenacoelomorpha. But when we talk about the synapomorphies of big groups and about animal design or the bowel plan writ large, that's often in chapter 4. So you'll keep coming back to chapter 4 throughout your time in this class. So our understandings, as that quote says at the bottom, about bilaterian relationships have undergone a major paradigm shift within the last decade. So the bilateria, this big red, the big blue box that I just surrounded in red, includes all of the animals outside of lots of you are familiar and kind of very friendly with corals. and other cnidarians, but really this is the majority of the species diversity of life, right? Our bilaterians. So we've got Ecdysozoa and Spiralia as two really species-rich groups because they include the spiralians, include the mollusks, and the ecdysozoans include the arthropods, and therefore the insects, and they're protosomes. And that's the other major group within the nephrozoa, along with us, with the deuterostomes. And our group, the craniata, which you took a whole course on, like imagine that, it's like spending a whole course on one tip of this tree. It's bananas to me that we have to then divvy up all of the rest of them here. Doesn't seem right to me, but our close relatives like the echinoderms, the hemichordates, the cephalochordates, and the urochordates. Now, that leaves this clade here, the Xenacoelomorpha. And that's essentially the question that we're going to talk about today, how our thinking has changed about where do the Xenacoelom, what are they? Where do they sit within the Bilateria? Are they a sister to all the other groups that we just talked about, the deuterostomes, the spiralians, and the ecdysozoans? Or are they a member of one of those three clades? So what do they look like, first of all? They're gorgeous. If you like small, flatworm-looking or empty coffee sock holding things. This is Xenacoelomorpha bacchi. This is another Xenacoelomorpha, and these are all aceals. So this is a group that was combined from two formerly separate taxa, and we will talk about kind of where we thought they sat in the past. But let's just kind of take a second and put up this great, really informative box of information, the characteristics or the synapomorphies of the Xenacoelomorpha, that goes into greater detail than the bullets that I will distill this down to. But they are soft body, dorsal ventrally flattened, so very flat things, almost exclusively marine worms. They're weird. They are a metazoan with unique pulsate bodies that are found in no other phylum. They have a ciliated epidermis that allows them to crawl through the world in a way that no other metazoan really does, that no other bilaterian metazoan does. They have a mid-ventral mouth and an incomplete gut. That is, they lack an anus. We're going to keep coming back to the anus here. Discrete organs, no discrete circulatory system, no discrete excretory system like protonephridia or nephridia, and no organized gonads. They're a disorganized, very simplified dorsal ventrally flattened thing. And they appear to have direct development, so there's no larval forms known at all. They do have circular and longitudinal muscles, so there is a muscle system. And where they sit phylogenetically has been called, you see there's a lot of purple hues here, where they sit phylogenetically has been called the mystery of the purple sock. I love that. We're going to talk about the purple sock by showing you like a little tiny white 1980s tennis sock. This is Xenacoelomorpha bacchi. This is a 25-year-old video that is still up on YouTube that a scientist named Max Telford filmed in Sweden. This is the sole member at that point known of Xenacoelomorpha, this ciliated dorsal ventrally flattened, very simplified metazoan of uncertain at that point phylogenetic affinity. Where it sat, who knew? This is my kind of distilling of some of those high points. The synapomorphies is that they have no gut, no anus, unique cilia along the epidermis, no discrete organs, no known larval forms, and are comprised of two very divergent groups. I've nodded to this already. The Xenacoelomorpha, which is this, and the aceals, which are some of these. Now, one of the cool things I'll add to at the bottom is that they also have, remember the statocyst from the other day with the ctenophores, this little ball that floats around, and its response to gravity and direction lets the ctenophore, and in this case also the Xenacoelomorpha, understand where it is in the world. They have one up at what you would call its front end, even though the mouth doesn't sit in its front end, it sits kind of mid-stomach. Okay. Apologies for jumping back and forth here. I'm quick on the trigger finger. So, diffuse nervous system, the statocyst, and a mid-ventral mouth. Now you're looking at two different live examples of that cilia kind of letting the purple sock inch around. That's what was just happening in the video on the left-hand side. And on the right-hand side, you have some micro CT scanning tomography of the midventral mouth, that kind of open pit there, of a recently described species from Japan. So not a lot of characters here, right? Lots of simplification. Really just like a pillowcase kind of moving around through the world. And what we see here, this is a classic kind of an old school phylogeny that appeared in the text that I took, and I show it to you just because we used to include our understanding of the Xenacoelomorphs being in two large groups, not monophyletic, two paraphyletic groups within the flatworms that we'll come to very shortly. So the flatworms are things like planaria, the free-living planaria, that's this kind of diagram here, and the parasitic monogeneans, the trematodes and the cestodes. So it's another whole phylum within the Bilateria. And we thought that this very unsatisfying hypothesis that Xenoturbella and the aceals were somewhere in there. Unsatisfying even to the people at the time who created this phylogeny, I can assure you. We know more now, so let's unpack where we think they sit. But let's talk a bit. I'm going to focus, the narrative today is going to focus on remember, Xenacoelomorpha comprised of two divergent groups, the Xenoturbella and the aceals. This is a little bit of a nod to the aceal because the rest of today we're going to zoom in on a character study on the Xenoturbella. But these are the aceals. So all those other traits, like very dorsally flattened, lacking lots of things, no mouth, no anus, lateral branches. They have some phagocytic glands that allow them to digest. And in this case, they do have some gastrovascular things. And you can see that there are eggs. And so a little bit of a sexual reproductive system and a slight hint of gonochory. So this key question, that's my nod to the aceals. We'll have to take this bigger course writ large when it's like 96% of your courses to get good time for everybody. But how will we focus our time? We're going to talk about how the Xenacoelomorpha alters our understanding of the phylogenetic systematics of animals. Where do they fit? They're in purple. So Dunn talked about this as two different hypotheses, or kind of distilled the phylogenetic thinking of the world, that the Xenacoelomorpha is sister to all the other Bilateria. That's this phylogeny here that I'm drawing my mouse over. So we've got Xenacoelomorphs occurring as the earliest or the first branching Bilaterian, and then we have to see the protostomes and then the deuterostomes. The other phylogenetic hypothesis is quite a bit different, and it suggests that the Xenacoelomorphs are actually a group within the deuterostomes. That's kind of bananas, because it essentially blows up this group here. So right here, see the nephrozoa? So the nephrozoa is a clade that's supported in your Dunn phylogeny by the idea that the protostomes and the deuterostomes all have excretory functions like proto or metanephridia that get rid of ammonia. And the Xenacoelomorphs don't have that. And so to place this taxon within one of those earlier stated ones, within the deuterostomes, says the nephrozoa is not really a thing. There's just Bilateria, and there's some groups where these characters have been lost. Casey Dunn and friends talked about it here. Only in the past 5 years that the union of Xenoturbella and the Acela received support from molecular data. And so these worm-like creatures without an anus also lack many other characters found in members of the Bilateria. So remember these threads that kind of flow through the sweater of this course, the currents that flow through the story of this course, the river of this course, is that we're bashing away at the idea that evolution is about the accumulation of new things. Evolution is often about the deletion and the simplification, and this perhaps is one of those stories. So the Xenacoelomorpha is it a bilaterian or is it a deuterostome? That's essentially the mystery that we need to solve, that we've been trying to solve as invertebrate systematists for the past 10 years. And I have some answers for you. But the first thing I'd like to do is to go backwards a little bit and talk about the history of this little fellow, Xenacoelomorpha bacchi, because it has had a tortured taxonomic history. If you think the uncertainty of whether it's a bilaterian or a deuterostome, that's like its current manifestation of its tortured taxonomic history, it's had worse. So, 1915 in Sweden, the western coast of Sweden, at a depth somewhere between 50 to 200 meters, Sexton here discovered this tiny little smidge of an animal. And it was described a long time later. And you can imagine my surprise when reading the description that it's nearly a 30-page long description for that little cashew of an animal. Why was there such a long time between the discovery and the description? Well, if you know a little bit about European history, between 1915 and 1949, Europe wasn't an entirely easy place to be, and certainly not a place to do things like taxonomy with great ease. And so this collection was made, and then it sat for a long, long time until it was described by Westblad in 1949. So the derivation of the word, turbella, so bella or turbella meaning worm-like, and xeno meaning strange. So that was the root of the word, a strange worm-like thing that's ciliated, that's very small, about 1 to 3 centimeters long. It has an apparent seems to be a diploblast, no mesoderm, a ventral mouth, no anus or distinct organs. So right away, we still have, like what they discovered then, these are some of the key characteristics that we retain and we know about today. Now, Sexton Bach, 1915, and Westblad in 1949, they were both platyhelminth taxonomists. So they studied flatworms. That was their bread and butter. And they named this a platyhelminth flatworm. Now, lots of those other characters that you see there are not characteristics of flatworms. And so I think this is probably a case of like round peg into square hole, or something that's called the rule or the law of the instrument, where if you're trained to work with hammers, what you see is nails. And so as a platyhelminth taxonomist, when presented with this very strange thing that doesn't possess a lot of the characteristics of the flatworm, they persisted in calling it a flatworm. This is a contemporary view of Sweden and the collection and the dredging of in the same area where this marine station was in southern Sweden. And this is a dredge, this kind of large cassette, kind of 6 foot long, like a Han Solo, kind of out of the carbonite. View of this is a Warren's dredge, and it's on a research vessel here associated with Gothenburg University. And that's how samples from the benthos from the deep ocean, beyond the reach of the easy scuba reach of for these people, are returned to the surface of the ocean. So that dredge is lowered down and then essentially dragged behind the ship. It's a really coarse way of sampling, and you can imagine that when they bring that up and they clean it off, as this researcher is doing with the hose, into the muck, they then have to search through this to find tiny 1 to 3 centimeter long, soft-bodied, ciliated animals. And you can imagine this is aspirational to think of it as a needle in a haystack. Would that it were so easy. This is the Xenacoelomorpha in a bucket of mud. So remember, the animal's soft and squishy and very hard to find. This is a very coarse way of looking for it. And so we don't see them a lot of the time, is essentially the end of it. And that might be a measure of the biology of the animal, but it might just be that we don't have a great way of looking for them. Now, in the early 90s, mid-90s, a lot of these animals had been collected in a way that allowed us to sequence them. And the advent of the sequencing revolution in biology, this was a very aberrant group, and so people were excited to extract some DNA and find where it sat phylogenetically. And so in 1997, there was a paper that was published with the first hypothesis of where Xenacoelomorpha sat. And you can see with this green, what am I talking about? Orangey screen, what I'm going to ask you to do is I'm going to show you, I'm going to annotate one little bit more and then pause me, okay? And answer this question. So based on the results of the sequencing and of this phylogeny, what is Xenacoelomorpha? And I'll show it to you in red. So what does the phylogeny say that Xenacoelomorpha is? Is that reasonable? Why or why not? I'm going to have a drink of tea. It's going to count down. You can restart me once you've made your statement. But please actively take part in this. Tell me what this is. All right. So what does this say that Xenacoelomorpha is? Well, it says it's a bivalve. It says that the thing that has no two parts and no valve is a bivalve. It's a mollusk. Now, we haven't unpacked yet some of the characteristics of mollusks, but it doesn't have a foot, doesn't have a radula, doesn't have a shell, doesn't have a mantle, doesn't have all of these things. This would be the most reduced, weird-looking mollusk ever. Now, there are some very simplified aplacophorine mollusks, and we'll talk about those in days and weeks to come. But this was a bit of a surprise, and hence why such a simplified phylogeny appeared in Nature in '97, the largest scientific journal in the world. So they used two different markers. One of them was the mitochondrial DNA barcoding marker, even though it wasn't called that at that point, and then a ribosomal marker 18S to propose that it was a mollusk. And there was a co-published article in the same edition that reported mollusk-like eggs being found inside the Xenacoelomorpha. And so they talked about this in the paper as being, this is not, Xenacoelomorpha is not the sister group to the Bilateria or any other, strike that word, basal metazoan or early first branching metazoan, but it's a mollusk. So it's a protostome. So it's within the Nephrozoa and it's lacking all of the features of the Nephrozoa. And that was published and their careers went on and people kept dredging off of Sweden looking for more and more of these things. Until, dun, 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 6 years later, sequencing technology had improved. And other markers had appeared in the common genetic libraries like GenBank, where people could compare and contrast what the sequences that had been collected. And there was some early thoughts that, well, you know what? I don't think that Xenacoelomorpha is actually a Xenacoelomorpha. And what they had sequenced was actually the meal. And those eggs that they had purported to find within the Xenacoelomorpha, those might have been eggs that the Xenacoelomorpha had eaten. So the sequences were real sequences, but they were of the meal. So they used data from 3 genes to show that the samples from 1997 had been contaminated or improperly labeled. And they were essentially showing that Xenacoelomorpha, what does it eat? Not what is it, but what does it eat? Their phylogenetic analysis took it out from, remember, so if we misconstrue that it's a mollusk, it sits in the protostomes. Their novel phylogenetic analysis now says this is not a protostome, it's a deuterostome. So they both agree that this is not the sister taxon to all the other Bilateria, but it's a deuterostome. So it sits related to hemichordates and echinoderms, so in our neighborhood, which is even more surprising potentially that this sock, this pillowcase of an organism, is right in our genetic neighborhood. It's one of us. So you can see here the Lophotrochozoa, so that's a spiralian group. In the Ecdysozoa, we've got echinoderms, hemichordates, and then sitting kind of as a divergent group, an early branching group there, the Xenacoelomorpha. And then us, the chordata. Us. I think that might be the only labeled phylogeny where we appear here. So this is kind of a banana, already a tortuous route. So 1915 collected, 1949 described as a worm, 1997 described as a mollusk. 6 years later, what is that? 2003, it's an aberrant kind of deuterostome. And they wrote an editorial. Henry G., who was the principal editor of Nature at the time and kind of a great writer and a witty fellow, said this about it. And I love these phrases, so I bring them back to you. So as we reflect on Xenacoelomorpha's newly exalted status as a deuterostome, That's tongue-in-cheek. There's no exalted. There's no this group is because it's us is better. It's worth recalling similar examples where Aristotle classified tunicates as mollusks, and the Amphioxus was a type of slug. These are big taxonomy fails if you do the hashtag and the meme. There's no shame in having once been classed as a mollusk because many of the best deuterostomes started out that way. So, as we bounce along together, some of you might be thinking as the dude does, what? So is it a deuterostome or not? So we'll call back. Here's a callback now to Casey Dunn's and groups and co-authors' final position and the remaining big questions in animal phylogenetics at a phylum level. Is the Xenacoelomorph sister to all the other Bilateria? And Andre Henol and all talked about this as saying an odyssey. So they don't as a torturous route. They described it as an odyssey through the animal tree of life. The enigmatic Xenotrabella bacchi seems to have settled down as part of the Deuterostomia, either as a sister group to the Ambacularia or as a sister to all deuterostomes. None of the analyses presented here find strong support. None of their analyses find strong support for putting Xenotrabella in the deuterostomes. And they place Xenotrabella with the aceals as a newly proposed phylum, the Xenacoelomorpha, sister to all of the other bilaterians. Not everyone agrees. And Felipe et al., this phylogeny makes sense in terms of the shared characteristics of Xenotrabella and aceal, such as the ciliary structure, diffuse nervous system, but it implies the loss of, they say various, but numerous deuterostome characters in the Xenacoelomorpha, including coelomic cavities, through gut, and gill slits. So lots and lots. But their insistence, their analysis is that the Xenacoelomorph is a kind of deuterostome. Now, the orange screen. So I'm going to ask you to claim, to make some decisions here, because the positioning affects our understanding of nerve cord evolution. Because we're involving the deuterostomes, and that's where we see lots of the proto-nervous system, the nerve cord. So we have the Xenacoelomorphs here. This is the first hypothesis. Sister to all other bilaterians. Pause me for a second and state for yourself, jot it down. Where would the nerve cord evolve according to this hypothesis? Okay, we're going to do one more. This is the alternate hypothesis. All right, we're going to do the same process. I'm going to start the timer, pause me, and so the timer is really for me and live speak here, but pause me, and where in this phylogeny does the nerve cord evolve? All right, restart. We'll compare and contrast on the same page. Well, it says here in the top, and it says here in the bottom, and then it's lost. So all sorts of consequences phylogenetically about excretory organs, coelomic cavities, nerve cords evolved after the Xenacoelomorph separated from the Nephrozoa. That's the implication of the first. And that is where the majority of current evidence sits, that it supports, and this is what you see in the Dunn et al. Phylogeny. And we unpack that as a story, as a history of science story, to show you, and just kind of lives through another example where phylogenies are hypotheses, and hypotheses must be revisited as we generate new evidence to see if, in case they are no longer supported. So science being about properly observing the natural world, accurately observing the natural world, and asking better questions, not necessarily saying this is the answer. 2016, 2 more papers were published in Nature in the same edition, kind of like If you were flipping a hard copy, they were back to back. And they were about this mystery of the purple sock, but they were actually about the description of the purple sock. Sweden, where Xenacoelomorpha is from 1915, so for 101 years, it was only known from that area south of the Marine Research Station in the Swedish coast, only known from the Atlantic. An entire phylum, only known from there. And then, with some remotely operated vehicles that were being run off of the La Jolla and the Monterey Bay, that slurp guns, rewind that again, I love this video. Because, so this is an ROV, remotely operated vehicle, that has the capacity to collect very subtly and gently. And you see that little pillowcase being sucked up there by the slurp gun? This is the first time that Xenacoelomorpha had been seen in those large fields of oysters, or of other bivalves, oysters and other bivalves, these socks littering the landscape. Purple sock, purple sock, purple sock. All of these purple pillowcases in and amongst the bivalve shells. And so what Greg Rouse and other researchers from the Monterey Bay documented was this genus in the Pacific for the first time, way far away from wherever we had seen it before. And remember how big Xenacoelomorpha was, 1 to 3 centimeters? So these are an order of magnitude bigger. These are giants. So in fact, Xenacoelomorpha monstrosa, capturing that in the species name, being 20 centimeters long, nearly a full meter stick. And the cool thing was about, so remember where Xenacoelomorpha had been found, not just in Sweden in the Atlantic Ocean, but between 50 and 200, so relatively shallow depths. These things that the ROV had sampled, not with that coarse sledge, but were with the very subtle slurp gun, all of these were living over 600 meters deep. Some of them as deep as 3,700 meters, so nearly 4 kilometers under the surface of the ocean. So the only way we could have collected them was with these ROVs and the slurp guns. And one of the cool things about it was that because they'd been collected with the intent, with like a priori decisions to try and sequence these things, there were 2 genetic clades evident here. One of them appeared to be for the shallow species and one for the deep species. So you can see there, the red box around some of these things. And they also all seem to be collected in and around mollusks, in and around bivalve mollusks. Not because they're bivalves, but because they seem to also like eating bivalves. So shallow and deep. So here you can see. So this is Baki. That's Sweden. This little, the kind of pale yellowy browny one here with the furrow right across the ferried furrow brow, right across the center. So there were 4 species described in this, and one is Hollandarum. This is shallow, so kind of 650 meters or less. But look at how genetically similar. This is the Pacific. That's the Atlantic. Very genetically similar. But then lying beside each other, essentially in a depth transect running off of the California coast, we see Monstrosa, Profunda, and Churro. So Monstrosa is big. Profunda is deep. Churro looks like a churro, and so that's what they called it. And so all of these things, and we're very genetically differentiated there. Let's look. We've spent some time with species descriptions, and you're kind of working on your species naming assignment. So this is churro. So there's the holotype. Where did it come from? This kind of fault off of the Gulf of California in Mexico, 1,700 meters deep, and there's the dive information. And what did I also want to call attention? Oh yeah, when they extracted the DNA, they found the same problem that had been demonstrated kind of 97 to 20 years earlier with co-occurring food DNA, exogenous DNA. So not contamination necessarily, but just exogenous DNA, not of the self, but of either of a thing that's living with it symbiotically, or in this case, the thing that it has eaten. So this is the bivalve that was present in the tissue. That's pretty exciting. The other paper, the Xenacoelomorpha as sister to all of the other Bilateria, talked about with novel sequencing and novel information, how this is really cementing this hypothesis that the Xenacoelomorpha, with Xenacoelomorpha as an example, this is not a funky deuterostome. This is not a thing that's in our neighborhood and with the sea stars and whatever. This is the sister to all of the other Bilateria. And this implies that the last common ancestor of the bilaterians probably was some kind of benthic ciliated worm with a single opening into an epithelial gut. So not an anus, but just it lost its byproducts across the cell wall. Excretory organs, coelomic cavities, and nerve cords then, in this hypothesis, have evolved after the Xenacoelomorphs separated from the Nephrozoa. And we've matched that on here. So there's the protostomes and the deuterostomes. There's the radially symmetrical cnidaria. Neither the cnidaria nor the Xenacoelomorphs have a really complicated nephridia. They're not bilaterally symmetrical. Well, the cnidarians aren't, but the Xenacoelomorph is. They have a mesoderm. The cnidarian doesn't. And so you see the Xenacoelomorph has some of the characteristics of the cnidarian, and then some of the characteristics of the bilateria, and then does not possess some of the characteristics of the bilateria. Kind of amazing. This is like recently enough that when the news, the mystery of the purple sock solved, this was big news on the BBC in the UK and the CBC in Canada. And the CBC radio program that you keep coming up against because I'm teaching this course, the flagship of our national broadcasting system, as it happens, tweeted this. This is 2016, so the Twitter was not a toxic place. The stuff of nightmares, a deep-sea mollusk with just one hole that looks like a purple sock. So I saw this, and I was offended. Or I was like, oh. And so I had a well, actually moment with the CBC radio as it happens, and I said, hey, whoa, not a mollusk. We've solved that problem. And they responded properly. And they said, thanks, Alex. Of course, we'll fix it. And then they deleted that tweet and retweeted this, the stuff of nightmares, a deep-sea creature with just one hole that looks like a purple sock. Well done, CBC Radio. Well done as it happens. Now, this is the observation from a student of a couple of years ago. This is Tina Belcher and Butts, and their observation was that there was so much poop content in this course. And that's, of course, in this course, we talk about the taxonomic breadth that includes the evolution of the through gut, includes pooping, because it includes a through gut from mouth to anus, and therefore poop. So much poop, so much butts. 2017, so a year after the publication of those two purple sock papers in Nature, there was another discovery made that talked about a new species from a new location, in this case named for where it was discovered, and it was discovered in Japan. And it was co-discovered, again, not co-published in Science or Nature rather this time, but two different publications that came from that event. And one of them was the fact that there's new evidence for supporting the purple sock as being the sister to all the other bilateria. New location, other side of the Pacific Ocean, so Sweden, California, Japan. They also discovered that a new species of orconectid that parasitize Xenoturbella. And this is kind of incredible because this is the first discovery. This is also the first parasite that we know about for this entire phylum of organisms. Now, what are the orthonectids? Orthonectids are, so there's, first of all, what are the orthonectids? Here's the look again of the scanning tomography. This is the farthest away from Sweden, but this is perhaps the most similar looking to the Swedish form of the Xenoturbella bacchi. This looks not like the purple socks off the California coast. And it can also be collected not so much with the ROV and the gentle slurp guns of the Mabari station, but with a dredge. And it shares with it the happy accident that it tends to live nearby to a marine biological station, so we're likely to bump into it. And so I place that out there just for you to consider, what have we not bumped into because they don't live near to a marine station, right? So, the orthonectids, quick aside here, remember we've talked about Xenacoelomorphs, we've talked about cnidarians, placozoans, sponges, and ctenophores. Orthonectids are in a big giant polytomy in the spiralia. we don't really know how many species there are. There's no estimate for there's not many of them. We know very little about the orthonectids, about an entire other phylum of animals, except they seem to be microscopic marine parasites. 20 to 30, fewer than 100 species described. They seem to parasitize the acials, platyhemionth flatworms, annelids, mollusks, nemerteans, so a good slew of the phyletic diversity in the ocean. And when they're in the host animal, they're present as a multinucleate plasmodium, which is so absent a whole bunch of things, almost looking like an amoeba. And the adults have a very simple morphology that lacks digestive system, lacks an excretory system, and lacks circulatory organs. When they're jettisoned from the host animal and live their juvenile life out in the open ocean, we know next to nothing about them. And so here's the plasmodium here. And then we've got a male and a female. Again, so males being kind of inconsequential, the females being necessarily larger because they contain the eggs. And then there's a whole, what do they do when they're out in the ocean? So this is another mystery that we just discovered packed inside of a mystery. This is like a murders in the building from a phylogenetic perspective. We'll come back if there's time to the orthonectids like later on in the course. Now, the Schmidt expedition, so this is the expedition, this is the foundation that funds a lot of the research on this ship. These are microscopes that are on board the ship that are dealing with some of the samples that have been brought up from the bottom. And we're going to watch this video together. There's the robot. That's Sebastian the robot.
About being on an expedition like this is we have a chance to find new creatures. So there's been one that I'm sure is new here called Xenacoelomorpha, and we found one here in Costa Rica roaming over the mud. We had an unusual find. We saw some strange tubes. Coming out of the end of the tube was a white proboscis looking structure, and then all over the tube were these beautiful yellow tentacles. I managed to get out the occupants of the tube and immediately recognized that it's a group of worms called amphiretids. The creature on the tube, though, is an enduring mystery. That single collection gave us two mysteries. One we solved, one is still going on.
See, this is not...
We often have images in our head of iconic creatures, and it would be great to see them. And one of the animals I've always wanted to see is a sea cucumber, Pelagiotheria natanatrix, is always pelagic, only found swimming in the water column. No one's ever been able to have them for DNA sequencing, and we found two specimens on this cruise. Sometimes you see things in the deep sea that are well known by other experts, but they turn out to be a surprise for you.
That's amazing. Let me scroll back 2 seconds. So Greg Roos is going to come up again in this course because he has led a lot of the discovery at a very, not just of species, and he's been involved in the description and discovery of species. He was involved in the Xenacoelomorpha that you just heard about, the purple socks, but he's involved in more. He'll be involved in some of the annelid stories, some of the hemichordate stories later on. But then there's this gentleman here. This is Dr. Jorge Cortez, who is a Costa Rican researcher, a marine researcher from the University of Costa Rica, who is also on the science advisor on the board of the Guanacaste Dry Forest Conservation Fund, which is an NGO in Costa Rica that I am also a science advisor for. That's where my research takes place. Jorge's is in the marine protected area, and mine is in the volcanoes and in the tropical dry forest, rainforest, and cloud forest. And Jorge was actually on the microphone when the discovery, so a new phylum for the nation, for the country of Costa Rica, and because it's recorded, because it's streamed, live stream, so there's Jorge on the shore, and let's share a moment of the discovery of Xenacoelomorpha for the first time in Costa Rica, as Jorge adds a phylum to the nation's fauna. That's his voice.
Okay, let me just check this pink thing.
That's the moment of discovery. Let me just check this pink thing. There's other people on the deck with him that are watching the screen over his shoulder.
Xenacoelomorpha. Oh, wait.
Wait.
Okay, we'll probably get one of those.
Look at that.
Uh-huh.
Yeah, let's get video of this. Greg will want video.
Yeah, we should get that cube out of the way.
The Greg they just mentioned is the Greg from the last video, the Australian voice. He's not on deck right now. He's going to arrive to see this discovery that Jorge has just made.
So,
Holy shit. Sorry. Is that loud? No.
it's moving. That's awesome.
Yeah, that's awesome.
Yeah, we gotta stop and leave out.
So fast.
Look at it, yeah.
This is super cool. Yeah. So this is super cool. Definitely a new species. We found it living at. There's only ever been one species before in Sweden until a few years ago, and then we found them living in California in Monterey, and the only one near here is in the Gulf of California.
Awesome.
And at 3,700 meters. So here we are, 1,000 meters. We got it.
Isn't that amazing? I love that. I love that moment. And so, here's the state of play right now. That we have Xenacoelomorpha baki, that we have Xenacoelomorpha churro profunda monstrosa, that we have Xenacoelomorpha japonica, and then now, as of this time in 2019, Greg Rouse and Jorge Cortez and the rest of the collectors on that expedition to the Schmidt Ocean have discovered another new species that has yet to be sequenced, but is going to be described as a new species. So a torturous odyssey, a phylogenetic odyssey for Xenacoelomorpha and the Xenacoelomorphs that has resulted in them being combined, the Xenacoelomorpha, that we no longer think of them as a protostome that was eaten. We now know that those protostome sequences were a function of being eaten, or what they had eaten, the mollusks that they prey upon, that we no longer think that it's a deuterostome and kind of an aberrant deuterostome that has lost all the characters of being a deuterostome. We think that this is the sister taxon to all the Bilateria, the mystery of the purple sock, and still more to be found, because we bump into them historically in Japan and Sweden when they happen to be near, and in Monterey, when they happen to be near research station, and now, so the Costa Rican example, which is yet to be described as one of the farthest examples discovered from a research station. What else remains for us to discover with this lineage, this sister to our lineage, the Bilateria? The mystery of the purple sock. Part of the mystery solved, but like the murders in the building, new seasons, and perhaps you'll be part of it. So I'll encourage you to be good to yourself, good to those around you, and we'll rejoin in the next session to talk about flatworms. And as we back out of the classroom today, in the acknowledging the mystery of the purple sock, I'll show you some of the ROV sampling video with the Arctic monkeys and knee-high socks. There's the slurp gun. And you can see the bed of the bivalves. And there's the little pillowcase. All right.